A center of excellence, a nurturing space for innovation, creativity, and academic freedom. This is the University of the Philippines, Los Baños. As a constituent university of the University of the Philippines system, UPLB is a leading national higher education and research institution in various niche areas. Grounding itself on the needs of national development, UPLB cultivates well-rounded and critical leaders who are ready to lead breakthroughs and innovations. Through its industrial and academic partnerships, UPLB propagates its gains to advance inclusive development in various sectors. An educational institution that upholds honor, excellence, and public service.
A center of knowledge, innovation, history, culture, and biodiversity. This is the University of the Philippines, Los Baños. UPLB found its home at the foot of the legendary Mount Makiling in Los Baños, Laguna, a short distance from Laguna de Bay, the country's largest lake. It has its beginnings in 1909 as the UP College of Agriculture, a tent school for a handful of students. A year after, the Forest School, or what was to become the College of Forestry, also set up its home in Los Baños. In 1972, the UP College of Agriculture and the College of Forestry, that had by then become renowned institutions, were granted full autonomy as the University of the Philippines, Los Baños. Instruction, research, and extension in agriculture and forestry in the country took root and flourished in UPLB. Through its strength in these two fields, UPLB established niches in various areas: biotechnology, engineering, veterinary medicine, natural resources conservation, environmental science and management, human ecology, food science, economics and management, public affairs, and development communication. It drew its strength in the arts and other fields of science, from harnessing its expertise and human resource in the biological, physical, and social sciences. Today, UPLB degree programs continue to dominate over its counterparts. Over the years, UPLB has developed graduate programs that continue to attract students far and wide, including collaborative programs with international universities in food security, biodiversity conservation, and development. To make graduate programs more accessible to professionals, UPLB offers them off-campus. Recently, UPLB established the UP Professional School for Agriculture and the Environment in Mindanao, the country's food basket, in order to help build the knowledge capital that the country needs to attain food security. Instruction in UPLB is enhanced by a strong and vigorous engagement in research and public service that enables it to produce knowledge and generate technologies and innovations that promote sustainable living, making it a strong force in the war against hunger and poverty in the country. Extension and public service are the mechanisms by which UPLB brings this knowledge and technologies to the people, as well as for it to know what they need and to help shape policies in the country. UPLB's reputation as a graduate and research university was formed through the continuous aspiration for excellence and the mutual reinforcement between instruction, research, and public service and extension. It brings in partnerships with local and international universities and provides mobility to faculty members, researchers and scientists, and students, and enhances their competitiveness. Instruction, research, and extension and public service in UPLB reflect the dynamism, honor, and excellence that its forebears set as a standard for its leaders to aspire for.
The world is in a constant state of change now more than ever. It's a world that requires resilience, flexibility, and innovation to overcome challenges and to pursue growth. That is why the University of the Philippines Los Baños is pursuing future-proofing initiatives to provide agile leadership and develop local and global communities amidst shocks and disruptions in an ever-changing world. Guided by compassion, an openness to working together, and the daring to seek out breakthroughs, UPLB constantly pushes for academic excellence, produces relevant research, establishes academic and industry partnerships, and cultivates creativity, critical thinking, and innovation. To keep up with the fast-changing world and to strive for inclusivity in development, UPLB seeks to level the playing field and expand avenues for acquiring new knowledge. It is reshaping the learning experience, enabling students and educators to continue with the learning process in vastly different learning conditions. It is expanding its academic programs and supporting initiatives to create a conducive learning environment. To address the growing complexities of modern problems, UPLB continues to break barriers across disciplines to produce cutting-edge innovations and solutions that serve the needs of various communities. It is taking on challenges in food security and sustainability and coordinating research efforts to direct the university's resources toward critical research areas, making sure that the university-generated knowledge can reach its intended stakeholders and be applied to real-life contexts. UPLB is dedicated to its commitment to contribute to public good and social welfare as it continues to work with the local government and the community to address their most pressing needs. Amidst its engagements, UPLB remains as one of the top regional and global universities and continues to be recognized in the country and across the world. As the national university, UPLB aims to be a steadfast partner in development, as a producer of new approaches that minimize the cost and impacts of future shocks, and by honing leaders committed to knowledge creation, innovation and cutting-edge research, and public service. This is UPLB's roadmap to be an impactful institution in a constantly changing landscape. Responsive, innovative, future-proof.
Happy Friday morning, everyone. Today is December 9, 2022, and Team LRC is back for a new set of learning sessions through our tutorials entitled Genius Ka Talaga, a tutorial webinar series on genetics. So, kumusta po kayo sa ating mga nasa Zoom space at sa ating mga nasa Facebook Live? So, nakita ko na bumati na po si Sir Renato Alonsabe. Blessed day po sa inyo, sir. Dito po sa ating Zoom space, kumusta po kayo? Nakita ko na ulit si Jay Luis Infante. Good morning sa iyo. Si Noemi Ramilo. Ito yung mga kasama natin for the past 2 sessions. How about the others? How are you? Kumusta po ang inyong umaga? Kayo ba po ay nakapag-breakfast na? Good morning po, Jenny May Decena. I hope all is well po sa inyo. Alright, so it's a rainy morning po here in UP Los Baños. But nevertheless, our hearts are filled with sunshine dahil malapit na ang Pasko, 16 days left at Christmas na po. And we will end this very fruitful week with minds full of knowledge on genetics. Na? So, good morning, Jay Luis. All right. So, kaya naman po sa mga nakatutok sa ating FB Live, magandang umaga po sa inyo. Let us know kung saan po kayo nanonood ngayon. Please comment them down below. And we have here Maricar Joyce Busel, Jeline Escolano Paraon, and Monica Nepomuceno Tino, who was watching with us via Facebook Live. Alright, kung wala pa po ditong inyong mga friendships, classmates, and colleagues, itag na po natin sila dito sa ating FB Live at i-share nyo na rin po ang ating video para sama-sama po tayong matuto sa umagang ito. As of this morning, we had a total of 328 registered participants for this tutorial webinar series. So we have plenty of students and teachers across UP UP campuses, across the UP system, as well as other colleges and high schools across the Philippines na talaga naman ay very much willing to learn and relearn lessons on genetics. So I hope on board na po tayong lahat. Time check, it's already 9:03 in the morning. Welcome to Genius Ka Talaga, a tutorial webinar series on genetics. So genetics is a subject that both interests and challenges students, both high school and college students, no? So which is why for this tutorial webinar series, our team prepared 3 sessions for us to fully appreciate and comprehend the different fundamental lessons or basics sa genetics. For our students here in UPLB who are currently taking Bio 30, no? Malapit na matapos ang ating semester. So talaga namang ating lubusin ang ating review session for this morning. So as part of the center's team learning sessions, we provide these sessions to serve as a review or remedial in preparation for upcoming exams or other assessments. No? Okay. So I am Joshua Michael Jonas, University Research Associate and Tutorials Coordinator of the UPLB Learning Resource Center, and yours truly will be our session's moderator. We are now on our 3rd and last session on parallel minds, applications in different fields of study. So today, we will be discussing about how is genetics applied in other branches of science, or how is it utilized in other components of biology perhaps, and how does it help improve and sustain the life of all organisms, no? So we will be tackling all of that this morning for our last session. So just a few friendly reminders for our session. Number 1, please prepare your pen and paper and take down notes and actively participate in the discussion. Number 2, keep your microphones on mute and cameras off unless instructed to avoid distracting the speaker and your fellow participants. 3, use the Zoom chat box properly and responsibly at all times. Please keep your comments helpful and considerate to the resource speaker, moderator, and to your fellow participants. Number 4, if you have questions, please feel free to write them down below our comment section or chat box and make sure that they are relevant to our topic this morning. And lastly, please do not forget to answer the evaluation form after the session. Your comments and suggestions will be deeply appreciated by our team. And please be reminded that the deadline of responses will be until 10 p.m. tonight. Okay? So for those who will not be able to join us today because you have another class to attend to, or perhaps you have another meeting, don't you worry. You can always watch the replay of our sessions on our Facebook page. We now have 106,000 strong followers on our Facebook page. So thank you very much for your continuous and overwhelming support. Likewise, you can also watch the recorded session on our YouTube channel. Just search UPLB Learning Resource Center on YouTube, wherein we have around 3.38 thousand subscribers, right? So please continue to like, share, and subscribe, and also click the notification bell to keep you updated with our videos. We would like to also invite you to connect with us on Twitter, TikTok, and LinkedIn. So good morning po kay Orlando Servandil. Good morning, watching from QC. All right. So kindly use our official hashtag for this tutorial webinar series, which is hashtag GENE-ius ka talaga, para mabilis po nating makita ang lahat ng videos in this series using that hashtag together with hashtag Team LRC. All right. So moving forward, before we proceed with our last session this morning, let's have another energizer, or perhaps our last energizer for this series. So if you have already attended our previous seminars or webinars, alam niyo na po kung paano gawin ito. Sa mga ngayon pa lamang po susubok, madali lamang po. First, open a new browser or a new window and go to www.menti.com. And second, type in the key code that you see on your screen, which is 6350-6105, and answer the two questions that I will be posting on our screen. All right, so let me share my screen for our Mentimeter. What was your favorite topic discussed in the past two sessions? No? So for the past two sessions, we discussed about heredity, basic chromosomal principles, and the functions of genes, or yung ating mga iba pang components ng genetics. Ano yung mga naalala natin? DNA versus RNA, central dogma. Ako hindi ko talaga maaalaga yung TEGI method ni Sir Eman, no? Kita mo, sir, talagang tumatak yun sa akin. Ano pa ba? We also did problem solving. No, yung ano ba yun? Yung sa when to use and when not to use yung ating Punnett square. Ano pa ba? Sa ating mga participants dito sa Zoom, ano ba ang inyong mga naalala from our past two sessions? No? Dapat at least makana tayo. 5 or 6 responses siguro. Sige nga. J. Luis, nakasagot ka na ba? Mutations. Ano ba? Christine, Dawn. Matatawag ako na para makasagot kayo. Jenny May, ano ba ang inyong mga naalala? Anne, Noemi, Chantel. Ano pa ba yung mga natatandaan natin? So, very fruitful yung ating mga napag-usapan ng last two sessions. No? Ating central dogma, translation, no? Yung ating central dogma, DNA versus RNA. Okay, sige. So let's proceed with our next question. No? Ito naman. What message would you like to say to Prof. Eman after this tutorial webinar series? So ano po ang gusto niyo pong iparating kay Sir Eman after being with us for these 3 sessions? No? Si Sir Eman ay very busy, especially he is currently preparing for his PhD studies next year, pero he was able to give us time for this tutorial webinar series on genetics. So ano ba pong ating gustong iparating kay Sir Eman after this 3-day tutorial webinar series? Sige nga po, nag-send natin ang ating mga heartfelt messages sa kanya. Thank you for the new knowledge daw, Sir Eman. So talaga namang, especially dito sa ating mga Zoom participants, so marami ang mga students at marami din ang mga teachers. So I guess many teachers also were able to adopt different strategies on how to effectively relay the information about genetics. Ano pa ba? Sige po. Let's wait for 2 more messages siguro. Thank you po. You are amazing, Lux. Amazing, Sir Eman. You made genetics enjoying. Aw. Iiyak na yan si Sir Eman. Thank you for the information. Oh. So talaga namang, kasi sir, di ba, kasi in genetics, usually it's one of the, kumbaga, kinakatakutan na branches ng science natin, no?
Yes,
Especially
sir.
pag nandun na sa mga formulas part, yung meron ng computations part, no? Pero ano, talagang it's also within the strategy of the teacher or the instructor on how to make it more fun and alam mo yun, more absorbing, especially with the many concepts na dapat nating ma-digest sa genetics. Ako mismo nung genetics ko nung high school, iniyakan ko 'yan eh. Pero anyway, saka na 'yan. Minsan nga yung iba umiiyak pa, sabi ni J. Lewis. So, true 'yan, no? Especially kaway-kaway pala sa ating mga Bio 30 students. Kumusta bang ating mga nakaraang 3 exam? So meron pa tayong isang exam na susunod, no? Sa Bio 30. Alright, okay. So yun lamang po siguro for our messages. Maraming salamat po for your heartfelt messages for our resource speaker. Okay? So I guess hindi ko na po papatagalin pa. Let's go now to the main part of our session. So I'll be introducing for the last time our speaker for this 3-day webinar series. So our speaker finished his Bachelor of Science degree in biology here in UP Los Baños and his MS in genetics. Ay, wait, tama ba, sir? Genetics ba ang MS niya?
Yes po, sir. Genetics.
Genetic, okay, tama. And his genetics is Master of Science in Genetics also here in UPLB. And he will soon be pursuing his Doctor of Philosophy. So hindi ko na muna isi-spill kung kailan at saan. But currently, he's an assistant professor at the Genetics and Molecular Biology Division of the Institute of Biological Sciences, College of Arts and Sciences here in UPLB, to share with us the different applications of genetics in other fields of study. Dear friends, let us welcome in the Zoom space Assistant Professor Emmanuel P. Galang. Sir Eman, good morning po, and you now have the floor.
Thank you, Sir Jonas. Sir Josh, I'll take it from here. Maraming salamat. So guys, magandang umaga sa ating lahat. We've been through a very rough journey. And the past 2 sessions have already equipped us on what really happens with the genetic material in the form of our chromosomes inside the cells. No? So let's have a brief review muna kung saan tayo nanggaling from our second session. Mag-share screen ako. 1 sec. There you go. So recall from our previous session na we focused this time on the genetic material. And apart from understanding what happens during mitosis and meiosis, specifically during cell division, nalaman natin that our chromosomes, the carriers and the conduit of genetic information, are nothing more but nucleic acids, specifically being DNA. So ayun. What makes DNA super special is the fact that it is the only informational biomolecule, whereas the rest do not have the capacity to carry genetic information with them that is transmitted from one generation all the way to the next. So nucleic acids are polymeric in nature. Ibig sabihin, these are composed of repeating monomers, and these repeating monomers are in the form of your nucleotides, composed of specific parts such as the phosphate backbone, the 5-carbon sugar or the pentose sugar, and the nitrogenous bases. This structure of the nucleotide is universal in our DNA, all right? And also in our RNA. So we have discussed also their specific characteristics and kung ano yung ambag nila sa ating DNA structure. We also discussed in the past ano yung difference between DNA and RNA, one being the more permanent type of information that is passed down from one generation to the next, that is our DNA found in our chromosomes inside our nucleus. Well, whereas your RNA are relatively temporary copies of whatever information that is needed by the cell immediately. And so characteristics of the DNA double helix is the fact that it is anti-parallel and double-stranded, running in opposite directions in the 5' to 3' and 3' to 5', so on, respectively. So we have also discussed kung ano yung nangyayari and the processes needed for the DNA from a double helical structure to transform into that of the chromosomes we see under the microscope. So this is the concept of DNA packaging. No? So from the naked DNA, it assembles around histone proteins, and these histone proteins produce more solid structures in the form of a solenoid, eventually forming your loop domains. And these loop domains will coil around each other to form the arms of the chromosome. Tapos, yung DNA natin, being the central informational molecule, need to be maintained and interpreted so that the information becomes very useful and is usable to the cell. And the process by which that is possible involves the maintenance and the interpretation of the genetic material. And this is only possible through the central dogma of molecular biology, where we spent most of the time from our second session. So we have DNA replication, the one that is responsible for maintaining the informational molecule from one generation to the next. Also, we have transcription and translation, which are the processes necessary to interpret and actualize whatever information that is found in the DNA. All right, so we have discussed that replication occurs during the S phase of the cell cycle. And on top of that, this process will occur in the entire length of your genetic material. Lahat-lahat ng DNA mo i-replicate during S phase. And the process will involve multiple proteins that will denature the DNA. Ibig sabihin, from a double-stranded structure, it has to be reduced to a single-stranded structure. And from the exposure of these nitrogenous bases, we get to incorporate their specific the additional bases by virtue of specific base pairing. So we have discussed as well the presence of a leading strand and a lagging strand in our replication fork, wherein a leading strand is generally produced as synthesis proceeds towards, or rather as the 5' to 3' synthesis proceeds towards the helicase or the replication fork, whereas the lagging strand, ang kanyang synthesis ng ating DNA polymerase is away from the replication fork. Hence, it requires multiple priming events. What else? So with this process, we found out that DNA replication is semi-conservative. Ibig sabihin ng semi-conservative is that a newly synthesized DNA is 50% composed of the original template sequence and a newly synthesized strand as highlighted in yellow here. So ayun, nalaman na natin that DNA is replicated. Kailangan niyang i-doblehin so that after cell division, each daughter cells will receive the same amount of the original content. So transcription naman is a process by which we produce transient copies of the genetic material that is in the form of RNA. Because remember that the DNA, which is our core genetic material, is found in the nucleus, and there is no way for the cell to access the information if not for the production of temporary transcripts in the form of RNA that can be exported outside the nucleus and can be used as a template for protein synthesis. Take note that one of the major differences of transcription is in the fact that not all sequences in the DNA are transcribed, because not all sequences are coding for a gene product. As compared to replication, na lahat-lahat ng sequences, nire-replicate natin. But not all sequences are transcribed because not all sequences are gene coding. So these gene coding sequences are actually referred to as transcription units. So these transcription units is comprised of specific regions that include your promoter sequences that serve as the binding sites for the enzymes necessary for transcription, and also control the rate at which transcription occurs, and also the main coding region, which will specifically serve as the template for the next process, and also for the terminator sequence that will signal the entire machinery to stop transcribing because it has reached the end of the transcription unit. So our main proteins or enzymes for the process of transcription involve your transcription factors, which help locate the RNA polymerase to the site of the promoter, because once the RNA polymerase is already in the promoter region, it can start transcribing. So the product of transcription are your RNA molecules, and these RNA molecules will be necessary for the next step of the central dogma of molecular biology, which is your translation. So sa process ng translation natin, guys, this is where we produce our polypeptide product, which eventually becomes functional proteins that is responsible for a trait. So after the process of transcription, you produce multiple types of RNA. So there are 3 types of RNA found in our cells that are necessary for the process of translation, whereas there are other smaller types of RNA necessary for the process of controlling the amount of gene products in the form of your proteins. So you have mRNA, which serves as the template. This is also known as the messenger RNA, the transfer RNA or the tRNA, which serves as the carriers of the amino acid to the site of translation, and also RNA, which complexes itself with ribosomal proteins that will serve as the site for translation. So sila to yung major requirements for translation. And all these 3 are a product of transcription. So here we have that, we see rather that the mRNA sequence is composed of consecutive RNA, and these RNA will serve as the template for translation. The template for translation occurs as a triplet in 3s, which will serve as the signal to specify which specific amino acid carried by a specific tRNA will be recruited as we elongate the polypeptide chain. So we see here a translation unit wherein we have here the ribosome, you have here the mRNA, and you also, since nasa end part na to siya, hindi na natin nakita yung tRNA being recruited. But this is how translation will look like. So eventually, you will produce a fully functional, not yet fully functional protein, but this will undergo specific processes to make it functional. Say, for example, after natin ma-produce yung polypeptide sequence, it will undergo correct folding and at the same time, cell targeting so that it gets to locate itself in the site, in the cell, wherein it is really needed. Okay. So, na-discuss na natin na from the genetic information, you replicate it to maintain it, you transcribe and translate it to read it. So, there are actually changes in the genetic material that occur as we go through life. So, these mutations can either be somatic or gametic. Kapag sinasabi nating somatic yung mutation, these types of mutations occur in the body cells. And since in body cells sila yung tipong hindi natin ginagamit for fertilization or the production of a zygote, these are not heritable. So say for example, if nagpa-araw ako, tapos natamaan ako ng UV rays. And then these UV rays mutated some bases in my skin cells. So does it follow na mapapamana ko itong mutation na to sa offspring ko? The answer to that is no, because the mutations occurred in the body cells and not in the gametes. So these are somatic mutations. Gametic mutations, on the other hand, are the types that will eventually be passed on to the next generation because of the fact that these are found in the sperm cell or in the egg cell, thereby making them heritable and transmissible from one generation to the next. So mutations are relatively weak, but these are the only forces in evolution that can introduce new alleles and new traits in the population. So variation is the raw material for mutation. So we have multiple types of mutations. So it can be as small as the type that will span a few nucleotides, or as large as the type that will span chromosomal segments. So let's take a look at nucleotide mutations. So nucleotide mutations, as discussed in the past, are the types of mutations that generally occur in the sequence level only. And this can involve insertions or deletions, which will eventually change the frame shifts of the open reading frames for translation. So insertions and mutation and deletions. So you also have substitutions. So in the event of substitutions, yung nangyayari sa atin dito is that you change one nucleotide into a totally different nucleotide, which might bring about a missense mutation that codes for a different amino acid, a nonsense mutation that instead of calling for an amino acid during the process of translation, it will code for a stop codon, or a sense mutation that will still code for the same amino acid, hence referred to as a silent mutation. So here, yun yung nucleotide mutations natin. What about chromosomal mutations? So chromosomal mutations naman medyo mas malaki yung span na kanyang kinocover. So because of the larger span of sequences involved in chromosomal mutations, usually these are the types that are more deleterious. So we classify chromosomal mutations as structural if it is in the structure of the chromosome that gets affected, or numerical if the mutation involves changing the number of chromosomes in the cell. So structural mutations can involve, number one, segment duplication, wherein a particular segment, as shown in the diagram here, makikita natin, say for example, a hypothetical chromosome with gene loci A, B, C, and D. Tapos in this event, dumoble yung segment that carries the loci for C. So sumobra siya ng isang gene. So this is duplication. You can also observe segment deletion if matatanggal naman yung isang segment. So you can totally imagine if A, B, C, D siya, tapos natanggalan siya kunwari ng B. So yung resulting chromosome will only have A, C, and D, thereby leading to gene loss. So yung problema, guys, with chromosomal mutations is that medyo mas malala yung effect niya kasi it's either you double the gene dosage or you reduce or totally remove a particular gene dosage, which is more harmful as compared to producing relatively less functional versions of the proteins, which we can observe in nucleotide mutations. So yun, so segment duplication and segment deletion. We also have segment inversions. Ano nangyayari sa segment inversion? So imagine that all of the sequences, or rather all of the loci in the chromosomes are occurring in a linear fashion. So if you invert a particular region in that linear sequence, so mangyayari niyan is that you will somehow disarrange the sequence as it originally was shown. So say for example, makita natin dito, so same chromosome A, B, C, and D. Tapos nagkaroon ng inversion. So naging A, C, B, and D. Okay, so that's for segment inversions. Translocations naman, makikita natin dito that this involves multiple types of chromosomes coming from different homologous pairs or bivalents. So say for example, originally we have here two different bivalents, one represented by the chromosome in purple and the other represented by the chromosome in orange. So makikita natin dito, after an event of translocation, nagkaroon ng interchange between the segments of at least one member of the two different bivalents. So yan siya. So chromosomal mutations. Whereas numerical mutations naman, this time, do not involve the structure, but we see here that it involves the number of chromosomes. We refer to it as aneuploidy if it changes the chromosome number. May dagdag or bawas in the chromosome number. Say, for example, trisomy 21, that is responsible for the Down syndrome. We observe here that instead of having two members in the bivalent for chromosome number 21, nagkaroon siya ng isa pang extra chromosome 21, thereby producing the Down syndrome phenotype, which is characterized by mental retardation and social intelligence stunting, so on and so forth, with additional physical manifestations as well. So you have trisomies, you have monosomies, wherein instead of having two chromosomes in the bivalent, nangyayari dyan is that you only have one. Actually, hindi ko na-mention in the past, but meron din tayong tinatawag na nullisomy. 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 Yung nangyayari naman dito is that you remove the entire bivalent. And this is generally not tolerated well in animals, but there are some plants that can exhibit nullisomy in certain bivalents. Euploidy, this time involves changes in the chromosome sets. So we are euploids in the fact that we are all diploids. And there are different types of ploidy levels, such as the monoploids, wherein they only have one set of chromosomes, triploids having 3 sets of chromosomes, and the triploid phenomena is the one responsible for the production of our seedless crops. Say, for example, watermelon, banana. And also we have tetraploids, which are common characteristics of wheat species, yung ginagamit natin for flour. And so these are generally tetraploid plants. So you notice, guys, that in numerical mutations, mas natotolerate siya ng plants as compared sa animals. Tayo parang up to trisomies lang ata yung kaya na i-tolerate ng animals as compared to plants that are more resilient to changes in the chromosome number, to the fact that they even change the entire set of chromosomes, say for example euploidy, so on and so forth. All right, so our mutagens are the ones that cause mutations, and these are classified as either chemical mutagens that our bodies are oftenly exposed to, as say for example diet or working hazards, or as simple as going out under the sun, or rather as simple as exposing ourselves to certain, let's say, vehicle smoke. So these are chemical mutagens. We also have physical mutagens. So these are forces in nature that normally occur, such as drinking very hot tea, or even exposure under the sun, which contains UV, and also ionizing radiation, such as the alpha, beta, and gamma radiation. Probably you've heard of this scare way back year 2010 or 11 or 12, wherein parang sinasabihan tayo na wag itabi yung cellphone as we sleep because it releases radiation. And this fear has been propagated very, very well and sensualized and romanticized in mainstream media to such a point that even my mom now believes that this is true. But there are studies that say that although phones emit radiation, these are not the types that will generally harm us. Yung pinaka-harm siguro is towards our eyes because of the intense glare of the screen. But it's not harmful at all. Kahit iunan mo pa yung cellphone mo, walang mangyayari sa'yo. So there. So these are induced agents, or rather these agents induce mutagenesis. So these are responsible for inducible mutagenesis. Whereas spontaneous mutagenesis also occurs. Ibig sabihin, nangyayari lang siya just because. These common examples of these are replication errors. Because as much as DNA polymerase, our main enzyme for DNA replication, is very, very faithful, and at the same time will always adhere to specific base pairing. But just like any other system in this universe, it is not 100% efficient and effective. It will always, these things are always bound to create mistakes. So the error rate in DNA polymerases can range to 1 times 10 to the 8th or 1 times 10 to the 7th. So medyo maliit lang yung chances. But considering the billions of base pairs na meron tayo, these chances are still observable. So actually, these mutations occurring during the process of replication are the ones that we see as culprits because for aging, because as the number of cell cycles progress and the number of cellular generations progress as we age day to day, mas nag-accumulate and mas nag-accumulate yung mutations. And these mutations somehow change and affect the cellular machinery and eventually leading to our aging process. So isa din siya sa tinitingnan sa aging. So there. So that concludes our second session. So I hope na-review niyo yung concepts. And looking into our third session this time, we will look at more advanced and probably more practical uses of genetics. Kasi pointless lang din naman if puro lang tayo konsepto tapos hindi siya natin na-apply. So the first application of genetics is relatively more, let's say, observable because we can see this in our families. So the first one of the major fields in genetics is what we refer to as human genetics. And in human genetics, nakikita natin dito the pattern and the mode of inheritance of certain traits or even diseases across very large across families. Remember, during the year 1500s, there was no DNA sequencing. There was no molecular biology tools. And the only way for doctors to track diseases was to look at the trend on how these were passed from one generation to the next. Hence, pedigree analysis. So eventually, with the analysis of these traits, human genetics was born, wherein we study how traits are passed on from one generation to the next. So these traits, I'll share my screen again, are very helpful in helping doctors during the earlier times of modern medicine to understand kung paano ba masacircumvent yung isang particular disease. So a pedigree is very helpful provided that a particular trait is not really rampant, but observable across large families. So let's take a look at a sample pedigree. So a pedigree will always contain shapes, and these will represent individuals in the pedigree. So these shapes can come in the form of a square or a circle. So a square will indicate a male and a circle will indicate a female. And apart from these shapes, the shapes can also be shaded. So another important component of your pedigree is your shade. So say for example, a shape can be fully shaded, which will indicate being affected, or a shape can be, say for example, a female here that is partially shaded, or paminsan-minsan nga pwede nga ganito yung kanyang pagka-shade, to indicate a person that is a carrier, or a person or a shape can be totally unshaded that tells us that the person is unaffected. So the shapes indicate individuals, the shade indicates afflictions. Okay, so shapes and your shade. And also, you will observe lines. So your lines indicate relationships. So a vertical line indicates parent to offspring na relationship. Parent to offspring na relationship. So this is also our marker that one generation has passed. Also, your horizontal lines naman indicate horizontal relationships that do not span multiple generations. So usually makikita natin to siya as siblingship or as a couple. So kung mag-asawa, nilalagyan natin, so say for example, if mag-asawa si male and si female, there is a horizontal line that indicates that. If may magkakapatid, may horizontal line, tapos may vertical line to indicate siblingship. So the horizontal relationship is brought out of parenthood. So ayan siya. So bali may shared sila na parents. Okay, so the lines also indicate the possibility of, say for example, twins. So kapag sinabi nating twins, makikita natin in our okay, so kunwari, you have here a twin. So an identical twin is referred to as this particular linear relationship na nagba-branch from one vertical line and may additional yan siya na horizontal line to really indicate identical twins. Identical twins. And also, twins can be fraternal if in the absence of one horizontal line dito na makikita natin sa bifurcation. Fraternal twins. All right. So here, say for example, probably one of the very famous family lines is the Lannister line in the Game of Thrones. Kung among you here watches Game of Thrones, sorry kung mas-spoil ko na kayo, but let's try to create an example. So, in the Lannister family line, which happens to be one of the major families of Westeros, yung nangyayari doon is that may tatlong magkakapatid. Yung tatlong magkakapatid, kung familiar kayo, sino nga sila? So, yung dalawa doon actually twins. Wait, let me erase this part here first para at least mas tayo. Mas may space tayo. So may tatlong magkakapatid and two of them are actually twins. So if you're familiar with the story, you know for a fact that these include Cersei and Jaime and their younger brother who happens to be an imp, si Tyrion Lannister, right? So this particular story is very central to the Game of Thrones plot. And then their mom and dad happens to be Tywin and Joanna Lannister. Ayun, so kung familiar kayo. And then, yung nangyari dito is that Cersei is married to Robert. And Robert happens to be the king. Ayan siya. And they said, or they claimed that their marriage has born 3 children. So, eto makikita natin si Joffrey, si Myrcella, and si Tommen. Ayan siya. However, I don't like to spoil, but nevertheless, I will. This is actually not a fruitful marriage because of the fact that these children are not actually King Robert's, but it is actually the offspring of Cersei and her brother Jaime. So therefore, this relationship doesn't exist at all. So makikita natin in the Lannister family line that we can actually draw a particular pedigree. So the pedigree involves individuals and also it involves linear generation. So we indicate generations according to the Roman Roman numerals that reflect which generation they are. So the first Roman numeral indicates the first generation in the trait in question. So the grandparents, the second generation, and the third generation is seen here. So makikita talaga natin na pedigrees generally help us visualize kung ano yung nangyayari sa isang pamilya. Ayan. So pedigrees are also very important in helping us identify kung ano yung trait or kung ano yung, or rather kung ano yung mode of inheritance for a particular trait. So what are our modes of inheritance? Modes of inheritance. So modes of inheritance generally is based on number 1. Is the trait dominant or recessive? So if the trait is dominant, generally we will observe that there is no skipping of generations. Whereas if the trait is recessive, we observe skipping of generations across very large families. So say for example, kung yung trait ay dominant tapos nanggaling na siya sa kinanununuan nyo, tapos it is generally the type that gets passed on from one generation to the next. Actually, one common example of this includes, let's say, hair color. Yan siya. And at the same time, recessive traits are generally the types na kunwari, ma-observe mo siya sa lolo at lola mo, pero never mo siya ma-observe sa parent mo. So it skipped one generation, tapos nag-manifest siya ulit sa inyong generation, sa current generation. So this is generally a recessive trait. So next is, on the chromosome, it is generally found. So it can either be autosomal or sex-linked. Kapag sinasabing autosomal, it goes to follow that these are found in your normal body chromosomes. So, ayan siya. So generally, this is observable if the trait, or rather, if in the pedigree, makikita natin that there is a 1:1 ratio between affected males and affected females. Ayan siya. So generally, the trait is autosomal if we see this trend. Or sex-linked, kung sex-linked yung trait, it means that the trait is found on either the X chromosome or on the Y chromosome. And if this is the trait, we usually do not observe a 1:1 ratio between affected males and affected females. So later we will see how these are observed in different pedigrees, which we will discuss today. So here, let's start with autosomal dominance. So autosomal dominance means that the trait is found in the autosomes and it exhibits a dominant phenotype. So let's take a look at this. So autosomal dominance, say for example, in this particular example, i-enlarge pa natin para mas madrawingan siya natin. Okay, so in this example, say for example, big A is affected and small a is not affected. So we indicated A as affected because this is a dominant trait. So let's try to identify the possible genotypes in this diagram or in this pedigree. So alam natin na those that are not affected will carry this genotype, right? Ayan siya. So homozygous, recessive sila lahat because these are all unaffected. Okay, whereas the ones that indicate a shade are generally affected. So we know that they carry a dominant allele, but we are not sure as to the second allele. So let's take a look at this particular case. So in generation number 2, this will help us identify the genotype of the male parent in generation number 1. So let's take a look at this. So this person indicates that this is affected, right? So it means that it got its trait from one of the parent, and that parent happens to be the male parent, as shown here. So therefore, alam na natin kung saan manggagaling yung kanyang pangalawang allele, and the second allele is of maternal inheritance. Bali manggagaling siya sa nanay. So since the mother can only pass on a recessive A allele, so therefore we know for a fact that this is a recessive A. So we know that the panganay of the couple in the first generation is a heterozygote for the trait. Okay, let's take a look at more examples in this particular pedigree. So makikita din natin na meron tayong anak dito with this particular genotype. Homozygous recessive siya. So homozygous recessive siya indicates that it got one of the alleles from the mother. So therefore, saan manggagaling yung isa pang allele? From the father. So therefore, this tells us that the grandparent of the of the individuals in generation 3 happens to be a heterozygote. So dito nakikita natin no, that the ratio, or rather, the traits on how they manifest sa mga anak will help us identify the genotype of the individuals concerned. So because of this, we have found out and identified that the parent, or rather the grandparent found in the first generation is actually a heterozygote. Ayan siya. So let's try to identify the possible genotype of the third female child. So first, second, third female child. So the third female child is said to be affected for the trait. And so saan kaya manggagaling? So since affected siya, meron siyang dominant na A na allele, which we can assume na namana niya sa kanyang tatay. Whereas, yung nanay niya will only be able to give a recessive allele. So therefore, making this individual a heterozygote in the process. Alright? So yan siya. Nalaman natin na heterozygote to siya. Oops. There you have it. Okay. So let's take a look at the bunso in the family, in the second generation. So nalaman natin that this person is affected, and this is contributed only by the father, thereby making the second allele come from the mother. Ayan siya. Oops, sorry. Wrong illustration. Misillustrated, sorry. To come from the mother. Ayan. So makikita natin this type of inheritance. Okay? So with this, can you guess what is the genotype for the first individual in the third generation? Can you guess? Sa chat. What do you think is the genotype of the affected female found as the first shape in the third generation? Tama. So the person here is assumed to be a heterozygote because nakita natin na affected siya, so matik may affected siya na allele, which must only come from the male parent. And at the same time, the second parent will only be capable of giving a recessive A allele. So makikita natin dito that this is an autosomal dominant trait. So let's try to verify if tama ba na dominant. Ano nga ulit yung characteristic ng dominant na trait? So a dominant trait will not skip generations. Okay? So will not skip generations. And at the same time, it is autosomal because the male to female ratio is generally on a 1:1 basis. So try natin count. 1, 2, 3. So 3 is to 3, which is in essence 1:1. Ayan siya. So autosomal dominance. However, guys, these rules are generally not applicable if the family is very, very low because masyadong mataas yung statistical risk. So say, for example, we have the same cross. So you have here an affected male and an affected female. So what if nagkaroon lang siya ng dalawang anak and these all happen to be unaffected? So by saying this, actually this is not a very good pedigree because of the number of individuals concerned. So usually pedigree analysis is only relevant if this involves very, very large families. So autosomal dominance yan siya. Okay, let's take a look at autosomal recessives. So autosomal recessives, makikita natin dito that there is the eventuality of a skipping of generations for the trait. Right? So let's use this. Okay, so N will be normal and a homozygous recessive will carry 2 small n's, which will indicate affected. All right, so big N for normal, small n for unaffected, small n for affected. So makikita natin sa first na generation na normal yung both parents, but we are not so sure kung saan, kung ano talaga yung kaniyang genotype. This can either be a homozygous dominant for the normal trait or a heterozygote for the normal trait. So let's take a look at this. So looking at the second generation, may nakita tayo na affected. So ano nga ulit yung genotype ng affected natin? Dalawang small n. So the small n allele can be contributed by both parents. Thereby, masasabi natin talaga that both parents are a heterozygote. Ayan. So may dito, naging informative talaga. Okay? So because of the affected individual in the second generation, this gave the genotype of the two parents in the first generation away. Okay. So with this, tingin tayo dito. So normal yung ating second individual in the second generation. So also normal din bale etong third individual in the fourth generation. So because of this, we can already deduce kung ano yung genotype ng two parents. Alright. So what about this one over here? So unaffected to siya. Ay, not affected pala, sorry. And questionable. So can you guess what will be the genotype of this individual? Of this particular circle? Take note that one of the parent is that the genotype is normal, or rather the phenotype is normal. So saan manggagaling yung normal? Sa parent na to. So saan manggagaling yung second yun na allele? In this particular parent, no? From this particular parent. So automatically makikita natin na heterozygote siya. So here we can observe the skipping of generations. So autosomal recessive siya in the sense that you observe a 1:1 ratio of the affected male and the affected female. And recessive siya in the sense that you observe skipping of generations. So you skip the first generation and the third generation, and eventually you manifest the traits in the second and in the fourth generation as shown here, making this an autosomal recessive trait. So relatively, I know you're bored with this. Let's go to something that is more challenging. So X-linked dominant traits are generally referred to as sex-linked traits because it is found in the X chromosome. So kung titingnan natin, since X-linked dominant siya, so pwedeng XA for affected, also X small a for normal, and Y for the other chromosome, of course. So let's take a look at this example. So eto guys, pwede nyo nang lagyan ng Y lahat ng males because these are known to carry the Y chromosome coming from the father. So in short, yung titingnan lang natin dito ay kung ano yung pinapamana from the mother. All right. So, all right, let's take a look at this. So the male in the first generation is affected. So yung kanyang X chromosome must carry an affected allele. All right? Whereas his spouse is unaffected, so it will have, or she will have, this particular genotype, indicating that she is normal. So with this, ma-identify na natin kung ano yung pwedeng mangyari sa next na generation. So the first offspring of the couple in the first generation is normal for the trait. So makikita natin na male siya, so therefore the male chromosome can only come from the father. Therefore, yung kaniyang second na chromosome will come from the mother. So yan siya. So therefore, yung kaniyang chromosomal constitution for the sex chromosomes involves this genotype. So it makes sense because unaffected siya because the X chromosome that will be inherited from the mother is generally a normal type. Okay, so tingin tayo dito. Let's take a look at the second daughter in this particular image. So sa yung since female to siya, so we know for a fact that this carries 2X chromosomes. So let's zoom in. So 2X chromosomes, 1X chromosome will come from the mother. So the mother will only give off one type of X chromosome. Yan siya. Whereas the other X chromosome must come from the father because female nga siya, di ba? Ayan siya. So the genotype of the second daughter of the first generation is a heterozygote for the X-linked dominant trait. So we can also assume that the third daughter of the first generation also has this genotype. So again, let's review. So one X chromosome will come from the mother, showing the unaffected trait, whereas another X chromosome will come from the father that indicates an affected trait. Ayan siya. So same is true with this. But pero meron pa silang isang anak na lalaki, no? So yung isang anak na lalaki nila will be, so yung Y chromosome came from which parent? From the male parent. And the X chromosome will come from the female parent. Thereby, since isang type na naman ng X chromosome yung pwedeng ibigay ng female na parent, so ganito yung kanyang genotype. So therefore, unaffected yung male na parent. Okay, so on your seats, can you try to identify the genotypes of the offsprings of the second female child of the second female child? So ito siya, guys. Highlight natin. Ito yung isa-solve natin. So what are the genotypes of these two children here? So let's start with the female na anak. So I'll give you time to work. Actually, guys, upon seeing the situation, malalaman mo kaagad yung genotype. Because remember, this is not affected. So therefore, yung kanyang alleles will be this. Tapos dagdagan pa natin ng X chromosome, right? So saan mga galing to? Ito, mga galing from this parent, whereas dito, manggagaling from the other parent, thereby making this very informative to indicate the genotype of the male parent for this particular individual. Ayan. So, mas napafind out na siya natin. Alright, so let's verify if tama ba yung ating assumption that this is an X-linked dominant trait. So, no skipping of generations. No skipping of generations. Nakita natin. And at the same time, X-linked siya. So, let's try to take a look at the ratio. So, the ratio is 1, 2, 3, 4. So, there are 3 affected males. And 1, 2, 3, 4, 5, 6 affected females. So this tells us that this deviates from the 1:1 ratio that we can expect sa isang autosomal na trait. So this tells us that this is X-linked or sex-linked. So you will also notice one trend for this particular example. And if you have observed that whenever a father is affected, automatically the female offsprings are affected. So this is one case. Also, observable in this specific lineage. So if the father is affected, the daughters are also affected. So in X-linked dominant traits, we generally observe a father to daughter inheritance. There, father to daughter inheritance sa X-linked dominant traits. All right, so clear ba sa atin, guys? So far yung topic, pulso ng masa. So far, so good? Guys, all right. Okay, so I'll take that. So let's take a look at another example. So X-linked dominant yan siya. So let's take a look at this. So this is very interesting because ano siya, X-linked recessive naman siya this time. Okay, so X-linked recessive, makikita natin dito na recessive siya in the sense that there is a skipping of generations observed for this particular example. So yung skipping of generations is observable in the second generation. So let's try first to identify kung ano yung ating gene notation. So X recessive means that yung trait is found on as a recessive trait. So X and will indicate normal. X small n will indicate affected. And Y will indicate the other chromosome. So let's take a look at this. So what is the possible genotype of the male parent in the first generation? So affected siya, no? So automatically, alam natin na may Y chromosome yan, right? So what do you think is the X chromosome for this parent? All right, so that's correct. So X small n Y. Let's take a look at the second parent. So this is a female, and it shows that this individual is normal. So normal, but we're not sure yet as to the second chromosome, if homozygous ba or heterozygous ba siya to normal. So let's take a look at this. So kung titingnan natin yung first male offspring in the second generation, let's try to deduce kung saan kaya manggagaling mga chromosomes niya. So yung chromosomes niya automatically contain a Y chromosome, right? Because this will come from the father responsible for his male characteristics. But ang sabi dito, not affected siya. So kung not affected siya, therefore, normal siya. So ganito yung kanyang genotype. Alright? So the genotype of the individual is XN and Y. So is this information helpful for us to help identify the genotype of this parent? Hindi pa. Hindi pa very informative kasi that can come from this chromosome, yes, but it can also come from this particular chromosome. So hindi pa siya super duper informative. So let's try to take a look at more individuals in the cross. Okay. So tingin tayo this time sa isang female na offspring. So yung female offspring na to, ano yung kanyang genotype? Yung genotype niya is, so therefore female siya, dalawang X chromosomes. So yung one X chromosome will come from the father. So the father can only donate this type of chromosome that carries the recessive allele. All right. So here, so what about this particular X chromosome which we can deduce to have come from the mother? So eto, so since normal to siya guys, so automatically normal yan. Okay. So informative ba siya as of this moment? Not yet. Hindi pa siya very informative. Kasi yung XN na to, na normal, might have come from this chromosome and not this. So hindi pa siya very, very informative. So moving on to the next generation, tingin tayo dito. So if we will look at this particular example, alam natin that this individual here is affected for the trait. So malalaman natin, yan siya. Okay? So looking at this further, so the Y chromosome must have come from this parent, and the X chromosome must have come from this particular parent here. So ano yung maging genotype nito? X big N, X small n. Alright? Kasi normal yung phenotype niya, pero nagkaroon siya ng anak na affected, and the affliction can only come from the X chromosome that she can pass on, because apparently the other sex chromosome came from the father, which is automatically a Y. Ayan siya. So taking a look at this further, we can see that there's a particular trend that we can observe. So, eto siya. So, saan kaya manggagaling yung ating... Okay. So, this X chromosome here can come from the father, whereas the other chromosome here can come from the mother. Okay. So, what is this story telling us? It means that there is not enough individuals in the population for us to identify the genotype of the female parent here. And that is totally okay. Because generally pedigree, for pedigrees to make sense further, they have to contain lots and lots of individuals. So the genotype of the parent, of the female parent in the first generation is inconclusive. Ayan siya. So inconclusive yan siya. So let's try still to identify kung anong type ng inheritance meron tayo. So we assume that this is X recessive. So let's take a look at the offspring. So 1, 2, 3, and 4. So 4 affected males is to 1 affected female. And also, so this tells us that this is X-linked and recessive siya in the sense that the second generation is skipping the particular affected phenotype. So ayan. So as much as possible, we don't want to see inconclusives in the family tree, but it is not avoidable. Or rather, sometimes it is not very avoidable because of the availability of the data that is present. But with these particular criteria that we have met, we can say that this is still X-linked recessive. Okay, so let's now take a look at the last type of inheritance. And this is what we refer to as Y-linked. So kapag sinasabi naman natin na Y-linked, the trait is carried in the Y chromosome. So automatically, if you have the Y chromosome, you will be affected right away. So the X chromosome generally do not matter that much for this particular trait. So kapag nakita niyo yung pedigree na lahat ng males ay affected in the process, then you can say that this particular type of inheritance is observable as a Y-linked trait. So Y-linked siya because all males carry the Y chromosome, which apparently carries the trait. So Y-linked yan siya. So here we can see that this particular trait does not exhibit dominance, no dominance, in the sense na yung Y chromosome natin will only occur as one copy in the pair that involves the sex chromosomes. Kaya hindi natin masasabi na Y-linked dominant or Y-linked recessive. Automatically, kapag may Y-linked ka, or rather, kapag may Y chromosome ka, you will express the trait. So yan siya. Okay, so on your seats, konting activity. So let's try to solve this particular problem. So the following pedigrees show the inheritance of the same trait in different families. Okay, so determine the mode of inheritance possible for this trait and symbolize the probable genotype of each individual in the pedigrees. Okay, so let's take a look at this example. So for the first type of the pedigree, can you guys guess what type of inheritance this is? May inheritance pattern ba kayo na na-observe? Is it possible for this to be an X-linked trait or a Y-linked trait or an autosomal trait? A clue, tingnan niyo yung ratio ng ating affected males and affected females. If it is close to 1:1, then that tells you something. What do you think? All right, I'll give you time to work on this. Okay, so may nagsabing autosomal dominance, says Elaine Isidro, and she is correct. So this is one classic example of autosomal dominance. Yeah, correct din si Hannah. Okay, next trait. Letter B. Ano yung possibilities? Autosomal recessive. All right? Ano pa yung ibang possibilities? Can you try to experiment if this trait can possibly be sex linked? Sige, let's try to explore other possibilities. Okay, so kung autosomal recessive, matik na may possibility talaga siya. Granting that this individual here is a heterozygote. Right? So kung heterozygote siya, matik na the rest of the individuals here are also homozygous recessives. So let's try to explore other possibilities. Is it possible for this trait to be autosomal recessive, or rather what if sex-linked recessive siya? If sex-linked recessive to, guys, so try natin. So kung recessive, X, Y, and this is X. Okay, so let's explore this possibility. So kung ganito siya, XN, XN. So kasi normal. Ay, sorry. So Y chromosome will come from the father. Ito siya. And the other chromosome will come from the mother. So since normal to siya, although we can already assume that these all contain normal na, okay, so kung X affected, ibig sabihin, so possible pa rin siya, no? Coming from the father. Okay. So in short, it's also possible that this trait is sex-linked recessive, no? May possibility din siya. So in short, example letter B is inconclusive. Okay, let's try some example letter C. Example letter C, let's try. Letter C is no longer inconclusive, you guys. Promise yan, hindi yan siya inconclusive. You will observe a particular trait. Sex-linked, tama. What gave it away? Ratio between the males and the females. Okay, what else? Correct? Oo. And makikita din natin na may very special na mode ng inheritance dito. Can you guess kung anong type ng sex-linked siya? Hindi siya natin matatawag na Y-linked because one of the affected individuals is a female. Okay. So let's try to take a look at this example. So for this particular case, wait lang guys, drawing ulit tayo. Sorry. Okay. So for this particular case, you will observe that the affected female is giving out affected sons. So ibig sabihin niyan, there's only one possibility to ensure that all affected sons are all the X chromosomes coming from the affected female will only give out affected sons. Makikita natin siya dito. Wait. It's now working. One sec. So i-drawing natin to siya. So if affected female, tapos affected sons. Okay. So this is actually sex-linked recessive na trait. Sex-linked recessive siya na trait in the sense that the female will have the affected allele, and the male here appears to be normal. So all males will have the Y chromosome coming from the father. And the X chromosome will only come from the mother. All right? So the only type of X chromosome found from your mother is X carrying the affected allele. So, yeah. So that explains why this particular trait is observable as the type that is manifesting a mother to son type of inheritance. There. So mother to son siya na type ng inheritance. Kung i-compare natin siya sa autosomal dominant sa sex-linked dominant, yung type of inheritance is a father to daughter inheritance. Whereas sa ating X-linked recessive na traits, mother to son naman yung type ng inheritance. Okay, so do I have questions coming from the class? No questions so far? Okay, so Sir Josh, quick break po muna tayo kasi before we proceed to the next topic. Relatively
Sure,
heavy.
sir. Okay pa.
All right. Maraming salamat po sa paghintay. So that quick break was very refreshing for us kasi our next topic is a relatively controversial one. And I know we have mixed feelings about this topic, but let's discuss it regardless so that we will have informed opinions whenever we are asked on the topic of genetically modified organisms. Kaya yun. So pinaghandaan talaga natin. Okay, so I'll share my screen, but first I'd like to hear your thoughts on GMO, and feel free to use the microphone if you'd like to air it out, or kung gusto niyo lang i-type. Wala din problema sa atin. Because, sige guys, go type as I keep on talking. Because there's this current running stigma with GMs, or with genetically modified organisms. I asked a lot of lay people kung ano yung opinions nila on the ethics of GMO, or genetically modified organisms. And a lot of them expressed fear. Say, for example, what is your initial reaction whenever you are confronted with the fact that what you're consuming is actually a type of product, a product of genetic modification? Say, for example, most of our cotton nowadays are a product of genetic modification to cotton plants that has been transformed to carry a gene that contains a that contains a gene that produces a gene product that is actually toxic to a particular type of bug, so that tataas yung production ng cotton natin. Kaya ayun, so there's this topic, or rather, there's this fear when it comes to the technicalities of the process. Now, what if what I'm consuming is actually producing cancer or causing people to have cancer? Kaya sobrang taas na ng incidence ng cancer ngayon. What if it's about genetically modified organisms? But for us to truly decide, usually the best decisions and the best convictions come out of deep research. So let's discuss what happens with genetic modification. So let me share my screen. So the concept of genetic mutations will first stem from the fact that there is recombinant DNA technology. Kapag sinabing recombinant DNA technology, this is simply the piecing together of different DNA sequences coming from different sources. Say, for example, I have a gene that I think could be very interesting, and I would want this gene to be present in a different organism. For example, I would want to produce tires that are generally glow in the dark. So what I will do is I will modify, or I will produce a genetically modified rubber tree that expresses the green fluorescent protein gene coming from, say, for example, from the jellyfish, so that all of its sap will, or rather, the components found in the sap of the rubber tree will contain the green fluorescent protein that is coded by the GFP gene coming from Achorea victoria, or a specific type of jellyfish. So in short, nangyayari doon is that you piece together a gene from a different source, that is the jellyfish, tapos pinasok mo siya sa genome ng isang plant, which happens to be the rubber tree, such that the rubber tree will start to consider it as one of its own, and eventually transcribe and translate that particular gene, producing the green fluorescent protein that is responsible for the fluorescence. Ayan siya. So here we discuss the concept of recombinant DNA technology. So one of the major contributions of recombinant DNA technology is that it allows expression of genes in different organisms. Prior to recombinant DNA technology, have you guys noticed that the price of insulin has drastically decreased across multiple decades? Way back 1980s, sobrang mahal ng insulin because the only source of insulin back then was for biochemists to go to, let's say, slaughterhouses or abattoirs. Tapos yung gagawin nila is that they will harvest the pancreas. And from that pancreas, doon nila kukunin via protein isolation and purification yung ating insulin. That process is very meticulous, very tedious, and will require the slaughter of many animals. And because very konti lang yung makukuha natin sa pancreas ng mga organisms or rather ng mga slaughtered animals sa slaughterhouses, that was the main reason why it was super duper expensive. So after the advent of recombinant DNA technology, they have somehow pieced together the gene that codes for insulin and nilagay nila yun sa mikrobyo. And the microbes now are the ones producing the insulin. And these microbes are being kept in a bioreactor such that they will constantly express the gene for insulin. And from there, we get to harvest insulin without the need for going to slaughterhouses and at the same time performing crude insulin extraction from the pancreatic tissues. Kaya sobrang nagmura yung ating insulin. So let's take a look at the process by which the process that involves the creation of a microbe or say for example an E. coli that produces insulin. So first, you have to understand the nature of your gene of interest. So your GOI stands for gene of interest. And gene of interest. And our gene of interest is the insulin coming from the pig. Okay. So yung gagawin natin dito is that, say for example, if this is our pig, so yan siya. Sorry. So if this is our pig, what we do is we get its DNA, and in the stretch of the DNA sequence, so sa sobrang haba ng DNA sequence niya, one of the genes that it codes for is the gene for insulin. So the advent of recombinant DNA technology came with the discovery of very special enzymes. And these enzymes are in the form of your restriction enzymes. What's so special with restriction enzymes is the fact that these are the ones capable of recognizing specific DNA sequences. And upon recognition of a specific DNA sequence, it will automatically create a double-stranded cut which enables us to cut a particular segment of the genome of, say for example, of the pig DNA. So makikita natin dito, say for example, if expand natin tong insulin na to. So this insulin, ito yung kanyang DNA sequence. Tapos ito yung kanyang transcription unit. Of insulin. So yung gagawin natin is that we have to identify kung ano yung sequences na present sa flanking regions ng ating gene, which happens to be insulin. Alright? So yung flanking regions natin are DNA sequences. And these DNA sequences, say for example, ATTCGG and ATTCGG. Kunwari, ito yung DNA sequence natin. And one of the sequences found in its flanking regions contains this particular sequence. So it was found out that your restriction enzymes have the ability to recognize a specific short sequence and create a cut in it. So, yung nangyayari dito is that say for example, one of these restriction enzymes, in this case, is EcoR1. Yung EcoR1 natin is a specific type of restriction enzyme that recognizes the sequence ATTCCG pala. ATTCCG. And once EcoR1, our enzyme here, recognizes this sequence, it will latch onto it and produce a double-stranded cut. So, yung mangyayari dito is upon DNA extraction of the pig's DNA, so sa ating reaction tube, you now have there the DNA, you will introduce EcoR1 or specific restriction enzymes that has recognition sequences. By the way, guys, when you say recognition sequence, or restriction cut sites, these refer to the specific sequence that can be recognized by your restriction enzymes, and upon its recognition, ikakat niya yan siya. So in this case, EcoR1 recognizes ATTCCG as its recognition site. So what scientists do is that they sequence the entire length of the DNA. So kapag nalaman na nila yung sequence ng entire length ng DNA, they will acknowledge certain recognition sequences or restriction cut sites such that they will be able to identify kung ano yung specific restriction enzyme na gagamitin nila to cut that particular sequence. So yung mangyayari niyan is because of the restriction enzymes, you will be able to isolate these specific genes. So you have restriction enzymes. So the birth of recombinant DNA technology came about with the discovery of sequence-specific DNA cutting tools in the form of our restriction enzymes. So there we have it. So now we have a means to cut and isolate specific sequences in the DNA. So ayun, pwede na natin siya makat. What now? The next thing that we are going to do is after restriction digestion. So tawag dito guys sa process na to is restriction enzyme digestion. There. So sa restriction enzyme digestion, it enables you to isolate a specific DNA length. And that DNA length in this example is the type that carries the gene for insulin production. So after restriction enzyme digest, you now have the insulin gene isolated from the rest of the genome. So yung problema with this is the fact that this is still naked DNA. So because DNA are generally fragile and have the tendency to be degraded by restriction enzymes, kailangan natin siya i-stabilize. And how do we stabilize our DNA? We stabilize this by inserting this to a vector. So what is a vector? So this is another important component of recombinant DNA technology. Your vectors are small. Sige, define natin yung vectors. Your vectors are small, circular DNA that has the ability to replicate itself, and the ability and specific parts that make it very functional. So later we will have a look at these parts of the vectors, specific parts. Okay, so yung nangyayari dito guys is that your insulin gene will be housed to a stable vector. So yung stable vector mo, remember, are small circular DNA structures. So DNA yan siya, double helical. All right. So here, this is an example of a vector that happens to be the location by which we insert our gene of interest. So yung insulin naman natin needs to insert itself to this particular location. So yung gagawin natin dito is that we have to open the entire vector sequence. But prior to that, let's take a look at specific parts of the vector. So vectors must contain a site for replication. So ibig sabihin, or also these are referred to as the ORDC sequence in the vector. This will signal or rather, this serves as initiation sites for the process of DNA replication. So ito yung ORDC sequence natin. So makikita natin yan dyan siya. Also, next to that are your selectable markers. Later, makikita natin yung importance ng selectable markers. But your selectable markers, by definition, enable us to, or rather, enable the organism that will carry the vector. Kasi later, ipapasok natin to siya sa isang organism. So it will enable the organism to be phenotypically distinct. Such that ma-identify natin later yung mga mikrobyo na napasukan ng ating vector. And also, it must contain a multiple cloning site and the multiple cloning site happens to be the site where the gene of interest will be inserted. Okay. So what happens at this particular point is kailangan natin i-open up yung ating vector so that your insulin gene will be able to find itself in the sequence. So what we do is we use, again, the enzyme. We use restriction enzymes. So these restriction enzymes will allow the opening of the multiple cloning site. So kapag na-open na yung multiple cloning site natin, it now becomes a linear sequence. And from this linear sequence na hindi na siya continuously circular, pwede na natin dito maipasok yung ating insulin gene. There. So this is now your insulin gene that finds itself in the restriction cuts, or rather in the cut site produced by restriction enzyme digestion of your vector molecule. So anong na-achieve natin dito? From the naked DNA, naipasok na natin siya sa isang vector that will enable it to protect itself from degradation and at the same time allow the sequence to replicate because of the presence of the RDC na site. Okay? So upon restriction enzyme digestion and the insertion of insulin, kailangan din natin siya i-ligate using the enzyme ligase so that the ends here will be sealed to the entire vector molecule. Okay? So the ligase ensures that your insulin gene is stably pasted into the vector molecule. So restriction enzymes. And then, so ano na ngayon guys? Meron na tayong vector molecule that contains our gene of interest. The next step in the process is referred to as transformation. The process of transformation will involve transforming organisms, well, in this case, a bacterium. And in the process of transformation, you will force this vector construct that contains your gene of interest na ipapasok natin siya sa loob ng bacterium. So, allows the vector to be housed by an organism. So ano nangyayari dito, guys? So transformation, so say for example, you have here E. coli na bacteria. So this is E. coli. And the challenge here is kailangan natin ipasok itong very small vector natin. Ipapasok natin yung small vector molecule natin that contains the gene of interest sa ating E. coli. Kailangan natin yan siya ipasok. Okay? So, how do we do this? Transformation will involve formation of transient pores in the cell membrane of the organism. So this is generally induced by exposing this to either heat or electric shock, which we refer to as electroporation. Upon exposure of E. coli to heat shock or to electroporation, what happens is that you produce transient holes in the process, and these holes become the site for entry of our vector molecule. So papasok yan siya. So upon its entry, yung mangyayari niyan, guys, is that you will now end up with an E. coli bacterium that now contains your gene of interest. So, or your vector that now contains your gene of interest. Ayan siya. So this is now a transformed E. coli. So anong mangyayari, guys, once it is already inside the cell? Once it is inside the cell, so kung ito yung E. coli natin, tapos ito yung vector with a gene of interest, it can now undergo the central dogma of molecular biology, and that involves replication. So in the process of replication, you will see that the copies for this particular construct will amplify because it undergoes DNA replication. Sorry. So guys, so dadami siya because of replication. Bakit nga ulit mare-replicate siya? Because it is a DNA sequence that contains the site, or rather the sequence that signals DNA replication, or that will signal DNA polymerases to gather itself in the ORIC region and proceed with the process of replication. Ayan siya. So, nare-replicate na siya. At the same time, nakaka-produce pa siya ng insulin protein So in producing the insulin protein by virtue of transcription and translation, we now have a mini factory that produces your insulin protein. However, yung problema with the process of transformation is that it is not 100% efficient. By saying that it is not 100% efficient, it means that in the experimental setup, there are some E. coli that will not be successfully transformed. Okay? So paano natin ma-select out yung ating mga successfully transformed E. coli from those that are not transformed? Remember that one of the major parts of your vector construct is your selectable markers. Its purpose will help us discriminate which are successful transformants and which aren't. So in the process, what we do is that we will try to grow this in a selective culture medium. So yung mangyayari dito, guys, is that assuming that we have different types of E. coli in the experiment na some are containing the vector construct while others are not containing the vector construct. So kung titingnan natin, bale, we will represent this as these three, wherein 2 are successful transformants and 1 isn't. So ano nga ulit yung ating selectable markers? So your selectable markers enable us to discriminate which is which. So a common example of selectable marker genes are actually number 1, antibiotic resistances. Say, for example, in this case, the selectable marker happens to be a resistance to the antibiotic ampicillin. So yung gagawin natin dito is that i-culture natin tong mga bacteria sa experiment natin kanina during transformation, and then we will grow this in a Petri plate that contains the ampicillin na antibiotic. So the ampicillin here will serve as the selective medium. So yung ampicillin, or rather as the selective agent, which we will add to the culture medium. So in the process, this will enable, or rather the selectable markers, since it confers resistance to ampicillin, it will allow the growth of colonies of these bacteria that happen to be successfully transformed. Ayan siya. Whereas this particular bacterium here that happens to be unsuccessful in the process of transformation will not be able to thrive in a culture medium with ampicillin because it does not have the vector construct that contains the gene for ampicillin resistance. This process enables us to specifically grow microbes that are now genetically modified. And the genetic modification the genetic modification and transformation of the vector molecule will allow it to carry with it the gene for ampicillin resistance, which enable it to grow in a culture medium that contains ampicillin. So with this process, you now have bacterial cultures that are successfully conferring and carrying the gene for insulin. So these are generally grown and cultivated in bioreactors so that insulin can be easily harvested from it. Ayan siya, so bioreactors. So we have now discussed kung paano nangyayari yung genetic modifications. So let's take a look at the process again. So the process of recombinant DNA technology involves, number 1, isolation of the gene of interest from the source material. So isolation of the gene interest from the source material will enable us to isolate a particular gene of interest. So isolating a specific gene of interest allows us further manipulation once it has been removed from the genome and is now present as naked DNA. So the gene of interest in its naked form is generally not stable. Due to the lack of stability, it has to be introduced to vector molecules. And these vector molecules will house your gene of interest because of its small circular nature. And its small circular nature contains specific parts that allow replication and selection. So replication is made possible of the ORIC sequence, which is also the specific sequence that initiates the process of replication. And selectable markers that make it phenotypically distinct so that later on in the process of transformation, ma-identify natin kung ano yung successfully transformed. Okay, so the first thing we do after getting the gene of interest is to get our vector and linearize our vector by subjecting it to further restriction enzyme digestion. Restriction enzyme digestion will linearize our vector, and upon its linearization, we now have the ability to insert your insulin into the sequence. So once it is already placed in this particular region in the multiple cloning site, we can now incorporate your ligase so that it can be sealed onto the vector. So your ligase enzyme makes sure that your gene of interest is properly sealed to the vector molecule. So you now have the vector molecule, and at the same time, remember that the vector molecule, apart from carrying the gene of interest and the ORIC sequence, it now has your gene of interest. Or rather, the selectable marker and the ORIC sequence, it now has your gene of interest. So, knowing that our vector molecule is now carrying the gene, we have to transform it. Or rather, we have to insert it to a living being so that it can be metabolized. So, yung nangyayari dito, guys, is the process of transformation. By transformation, what we do here is that the vector molecule is forced to be inserted to, say, for example, in this case, the E. coli bacterium, by inducing the formation of transient pores in the cell membrane of the E. coli. So heat and electroporation will allow these transient pores and membrane in the membrane to form, thereby acting as a passageway for our vector construct. And knowing that the process is not 100% efficient, what we have to do now is to perform the process of selection. So sa selection, this enables us to specifically grow E. coli bacterium that contains your gene of interest. So the vector molecule. So yung mangyayari dito, guys, is that we will now grow this in a selective medium that contains a particular antibiotic, wherein your selectable marker, or rather wherein your vector construct contains the gene for a selectable marker to confer resistance against that particular antibiotic. This gives rise to colonies that are successfully transformed because of the selectable marker ampicillin that gives them an advantage over those that are not successfully transformed. Okay, so this will then be routed to bioreactors and grown in bioreactors so that this will serve as continuous source for our gene product. Okay, so I would like to open the floor for possible discussions because this particular topic contains possible ethical issues. And say, for example, what is your take on genetically modifying higher organisms, say, for example, plants and animals? These are a cause for, or rather, these are topics for conversation. And at the same time, would you subject human genetic modification such that it provides us, say, for example, with resistance against certain pathogens or certain diseases such as HIV? Or would you go that far in terms of producing, let's say, a particular, or rather inducing a particular phenotype in babies, say, genetically modifying humans to be smarter, to be stronger, so on and so forth. So these are common concerns that generally involve the process of genetic modification. So despite these fears, actually, there is an ethics committee that screens all of the experiments that involve genetic modification. And prior to a scientist pursuing a particular experiment that involved genetic engineering, this has to be approved by an international body. So ayan siya, guys. I would like to open this floor and this time for conversations that involve genetic modification. What are your thoughts on genetic modification? And what are common misconceptions you think that can be clarified in this conversation? Okay, guys, the floor is open.
Siguro po, perhaps someone would want to raise their hand.
Mm. Mm.
Meron pa. Hmm. Baka speechless pa sila, sir.
I understand kasi genetic modification itself is let's say very controversial, and it took quite some time for humans as a society to actually embrace it. It has its pros, it has its cons, and right now there are even more sophisticated gene editing tools such as CRISPR and such as talens and zinc finger nucleases that, apart from simply inserting genes of interest to a particular organism, hindi lang siya basta-basta na makakakuha ka ng sequence, but rather this gives you the power to alter your very own genome without the need of incorporating a particular DNA from a specific source. So it's just like a tool that allows everybody to, or that allows the scientific community to rewrite the genetic code, or rather to rewrite one's genetic sequence. So from inserting from inserting and manipulating and piecing together different sequences of DNA, we have now come to this point na pwede na natin i-edit mismo yung sequence, like specifically editing or changing a particular base pair, so on and so forth, that can affect the phenotype. So, ayun. So, do I have questions, guys, coming from the class that concern this particular topic?
Ako po siguro, sir. Sir, ang question ko po siguro with regards to, especially with GMOs, with the concept of using humans as the test subject, what
Yes.
are the ethical, from your experience, what are the ethical considerations that should be taken into place? Kasi I think that's one of the number one things that those who are against GMOs content, yung ethical concerns. Is it right ba to make use of human subjects as a test for these experiments? Parang ganun, sir. What's your take on that? Or perhaps you have an experience with that, sir.
In one of our discussions in my MBB ethics class, there was this particular scenario wherein may isang Chinese scientist. And then what he did was that, gist lang, general. What he did was he genetically modified human babies. And then yung rationale ng study niya dun is that may ginawa siyang mutations dun sa receptor proteins specific for HIV. In turn, the babies will produce proteins that are defective in terms of these receptor proteins, such that yung virus will not be able to latch to it anymore kasi nag-mutate na siya, so therefore nag-iba yung kanyang protein structure. So, lo and behold, these babies are indeed HIV resistant. But the topic with ethics, just like anything else, is the issue on consent. Parang, kasi if the babies weren't given the chance to decide for themselves if they would want it for themselves. So that is one thing with ethics. It is the issue on consent. Say for example, ikaw Sir Josh, if ever magkaka-baby ka in the future, and that baby is designed to be super duper smart mathematically, and that can be achieved by virtue of genetic manipulation, kunwari, we have reached that kind of technology. But pinanganak, and eventually pinanganak yung baby, lumaki yung baby, matalino siya sa math, truth be told. But what if what he wanted or she wanted was music, was arts, but that particular choice was taken away from that person? So that brings about the topic of consent in genetically modifying organisms, specifically humans, to confer a specific trait, because they were never given a chance to choose for themselves. Because once the genetic modification is there, it's almost impossible to reverse it. Ayun. So that's one. Also, another problem with genetic modification is that just like any other experiment, it is bound to fail. Kaya, say for example, the Golden Rice Project, which is a genetically engineered rice that carries the genes for beta carotene synthesis coming from mais. Tapos, it took quite some time from the conceptualization of the experiment all the way to it being released in the market kasi they have to make sure that it is safe for food and feed. Kaya sobrang tagal ng mga proseso that involves releasing into the market these particular genetically modified organisms. Truth be told, we have a lot of researches and experiments at UPLB that involve these particular experiments. And a lot of our esteemed colleagues and faculty members have worked on, say, for example, Dr. Evelyn Texon Mendoza worked on a delayed ripening papaya and a particular resistance to a specific disease in papaya. So these are genetically modified organisms. Meron na siya, present na siya, and na-actualize na yung concept. But as of this moment, it's very hard to release it in the market because it has to undergo stringent processes. Kaya this leads me to another concern in genetic modification. It is the consumption of these genetically modified organisms. Are these safe for food and feed? The answer to that question is yes. They are 100% safe for consumption. What are the common fears ba if a person ingests genetically modified organisms? One common fear there is sabi nila na, hala, magko-consume ako ng GMO. Baka magiging GMO din ako. Kasi GMO yung kinain ko and they say, you are what you eat. But the truth to that matter is, number one, if you ingest, let's say, if you ingest a particular tissue ng plant that contains genetically modified sequences, there is no way for the DNA ng tissue na yun, ng ninuya mo, to incorporate itself in your very own genome. Kasi sa stomach acids pa lang natin, macha-chop-chop na yan siya. And there are no specific enzymes that will enable it to induce genetic modifications in our own cells. And also, so in short, there is no fear in that at all. Say, for example, kumakain tayo ng malunggay. And malunggay also contains its very own DNA. Bakit hindi tayo naging color green? Kasi mag-incorporate yung DNA ng malunggay sa atin. So as much as regular DNA cannot find itself in our own genome or cannot incorporate itself in our own genome, the same can be said true with engineered DNA sequences. So there is no way for GMs to cause GMs. Ayan. So that's one fear addressed. Ayun. Next. May tanong ata si Ana G. Namok. Sabi niya, Sir, can GMO fight against allergies? It's a possibility, yes. If it comes to that point na super advanced na yung society natin, we can produce organisms or rather crops that do not contain these allergens anymore. Or pwede naman tayo na i-engineer mismo yung organism to not contain specific receptors to these antigens. But etong isipin natin, anything and everything is possible once we fully understand the genetic code and once we fully understand the genome and how it works. Because in understanding a particular thing, you get to fully maximize it. And we know that since DNA holds the instructions for life, we can reach that point that we can alter it to such a way that we can achieve a particular goal. Let's say a cancer-free society, let's say an HIV-free society, or even an aging-free society. But that will take hundreds and hundreds, if not thousands of years from now of understanding the genetic material. But these can all be achieved by studying the genetic material. Kaya once, 'di ba, the DNA sets the rules for life. If you understand the rules fully well, you can reach to that point that you can tweak it and alter it to your own advantage. But then again, ethics will always come to play. So ayun. So allergies man yan, sakit man yan, it's possible. It's always possible.
Thank you,
Okay,
Sir
so
Eman. May
ano
iba
pa
pa
yung
questions?
specifically with the ethics of GMO? Kasi maganda siya pag-usapan. Lalo pa, we are educators. A lot of us here are educators. And it's really heartbreaking sometimes to hear my students dito sa UPLB tell me na, "Sir, sabi sa akin ng high school teacher ko, kapag kakain ako ng GMO, tutubuan ako ng pakpak. Kasi GMO yun." So it's best that we address these kinds of confusion and propagate the truth instead of propagating fear. Because in understanding something, we tend to appreciate the beauty of it, and at the same time, um, in its own understanding that particular thing, we also educate one mind that will eventually echo whatever you said. Ito, in the example a while ago, the process used ampicillin. Just in case the GMO gets inside the body, does it mean one will be resistant to ampicillin? Okay, so balik tayo sa fact na yung ampicillin will be yung ampicillin, bakit ampicillin resistant yung ating genetically modified bacteria kanina? Because because it contains the gene for ampicillin resistance. So yung ampicillin resistance na yun is in DNA form. But once inside the cell of the E. coli, it will produce the protein product that is responsible for ampicillin resistance. So once that particular gene will find itself in the human body, or rather, once, kunwari naka-ingest ka nun ng microbiome yun, there is no way for that to incorporate itself in any of your cells. So, hindi siya mapapasok sa genome mo. Hindi mo rin siya ma-express. You also do not expect yourself to be ampicillin resistant because it will not find itself in your genome. Okay?
I
Claro,
think I think Sir Eman related po, may nag-send po sa akin ng question dito po, no? Kasi ang question niya po is, if livestock, specifically chicken and other domesticated animals, eat genetically modified grains or feeds, will their meat, once slaughtered, will be also considered as GMO? I think connecting pa dun sa na-mention niya kanina.
So hindi siya GMO yung karne ng manok na kumain ng genetically modified na crop. Because hindi maa-alter yung DNA ng manok in the process. Kakainin niya lang yung crop, tapos kapag na-ingest na nasa tiyan na niya, i-chop off lang yun ng enzymes and ng acid sa tiyan, and eventually it will be nothing more but reduced biomolecules that will never find itself in the genome. Never. Again, I repeat, GMOs will not make you a GMO or will not make anything that consumes it a GMO.
Thank you, sir. Ito po, I have more questions with me here.
Yes,
Sige
opo,
po.
let's open
Ito
the
po,
floor.
sir. Ano po, sir? Next question po is, do GMOs have a direct impact on the environment, like in the energy flow, the carbon cycle, or the, ano yung sinabi dito, yung sa carbon footprints, parang gano'n, may effect ba po on GMOs? Or does it alter in any way yung mga ganong natural flows or cycles in the
Natural
environment?
processes. Actually, the answer to that question is no. Because remember, the DNA will only produce the protein, and the protein itself will be utilized by the cell. And once hindi na yan siya gagamitin ng cell, i-degrade niya yan. Hindi yan siya maiiwan. Because at the end of the day, kung ano man ang pinasok mo na gene, na organic material, magpuproduce yan siya ng organic material in the form of your RNA and eventually in the form of proteins. And these will all be easily degraded and not hamper any process in, let's say, for example, in the carbon cycle, so on and so forth. Proteins lang din yan, guys. You can imagine it like, just like, parang kuko lang yan, protina lang din yan, parang buhok lang din yan, protina lang din yan. It's just protein produced in a different source. Okay? Sir,
Thank you, sir.
why
Ito, may
GMOs
question
can
po
be
si
easily
Jay.
digested by our body? What is the characteristic of GMO that makes it easy to be digested? GMOs are digested just like any other stuff na kinakain natin. Wala siyang special na characteristic na mas madali siya i-digest. Point is, madadigest pa rin yan siya because these are all organic matter. Okay. So, sir, can dominant alleles change to become recessives? How scientists figure out which allele is dominant and which is recessive? Thank you for the question. So, the process of mutation can actually change. Kasi remember, when you have a dominant allele, it will code for a protein product that only one copy of that is enough to express the trait. Say for example, if the dominant allele, which is DNA in form, DNA yan siya. Tapos i-change mo yung certain sequences dyan, or i-mutate mo yung certain sequences dyan, it can ultimately result to a gene that is very similar but can have different characteristics. And that can be by virtue of dominance, kung mas naging mahina ba yung trait, or a totally different allele at all. Say for example, the eye color sa ating mata. So in short, the answer to your question, yes. Generally, the formation of recessive alleles are a product of mutation of the original DNA sequence. Ayan siya.
Thank you, sir. So I have another question here with me. Ito medyo interesting na pero parang leaning towards botany to eh. Ang tanong niya po is, can GMO crops contaminate neighboring non-GMO crops in a single field? So kumbaga parang ang ang question niya dito, sir, is for example, let's say there is one specific crop plant. Tapos 'di ba, meron tayong kino-consider yung pollination, yung ating mga what if yung isang GMO plant is yung pollen niya, yung kanyang fertilizing cells ay na-pollinate yung isang non-GMO plant. Will the be the product of that will be a GMO? Parang ganun po ang yata.
That is what you call environmental introgression. And that's a very good point. Kasi yung mangyayari niyan, the possibility of genetic contamination is there. Say for example, kapag kapag pinatubo mo yung isang plant na genetically modified, tapos pinatubo mo siya sa pinatubo mo siya sa tabi ng isang same plant that is a wild type or not genetically modified. Yung nangyayari dyan is that there's a possibility for the two genomes to mix sa pagmix ng kanilang gametes. So that is one of the main reason why genetically modified organisms need to be grown in a confined field testing facility so that these do not contaminate, or rather the genomes or the genetically modified genomes of these organisms will not mix with our wild type, because as much as possible, we would also want to preserve the genetic integrity of our wild types or our non-mutated ones. So ayun siya. It is sa bioethics, palaging nagkakaroon ng separate plot, paminsan-minsan fenced pa nga yan ng malala, and with particular restrictions in terms of the number of meters to the next life that it can possibly find, such as another plant. So yun siya. If you go to the confined field testing facility sa International Rice Research Institute, dun mo talaga makikita na very strict talaga sila dun. What goes in there, or rather what grows in there, must not go out, not unless it has already been approved by the government. Say for example, mag-move ka lang ng isang halaman na genetically modified from one lab to the neighboring lab, you will need a permit. Ganun siya ka-strict. Ayan. So, okay. GMOs are organic. I thought it's artificial. Okay, guys, I think we need to change our definition of what is organic. The term organic means that it is carbon-based. So say, for example, my hair is organic. My uh, my skin is organic because these are all composed of carbon molecules. Say, for example, if I will have a genetically modified gene produce the protein, it is still considered an organic molecule because these are still composed of long chains of carbons. Ayan siya. Kaya kapag very bad marketing strategy talaga yan kapag sinasabi na it's organic, parang ganun, parang say for example, this is organic milk or organic bread or organic organic whatever. Point there is anything in life that comes from a living organism is most possibly organic, regardless of it being genetically modified or not. So I think the antonym to GMO is not organic because GMOs are organic. The antonym to GMO is simply non-GMO. And there's nothing wrong with that at all. Ayan. So, sir, how can we use the full potential of talents? May nagtanong dito, sabi niya, talents. Yung talents is one of the earlier versions of our genetic modification tools. It stands for transcription activator-like effector nucleases. Tapos, yung ginagawa niyan is that this is one of the earlier tools for genetic modification na iibahin mo yung sequence. Parang genome editing tool siya. Just like CRISPR, but it works in a very different way. The full potential of talents is not actually being parang eto, my known experience with talents is that it's very difficult. And at the same time, it's prone to, let's say, experimental errors. That's why upon the surfacing and the advent of CRISPR technologies, almost all scientists turned their back against talents. Because now comes a more efficient, a cheaper, a higher success rate technology in the form of CRISPR-Cas9. Ayan siya. Sir, how does deletion mutation affect gene expression? Can you cite an example? A deletion mutation in the sense that you delete the entire gene? Noemi? Or you just delete a particular nucleotide from the sequence? Kasi, sige, let's discuss the two possible scenarios. Ay, teka lang guys, may time pa ba? So last 4 minutes.
Go lang, sir.
Okay. So a deletion mutation, generally, a particular gene contains deletion. Okay. So if a gene contains deletion, it results to frame shift mutations, di ba? So changing the codon or the reading frames eventually will bring about a series of changes to the amino acid sequence. And changing these will, remember guys, that if you change something in the amino acid structure, in terms of its amino acid sequence, there's a possibility always that it will affect the resulting protein product. And once that happens, it has the possibility to affect the phenotype as well. So these deletions bring about a change in amino acid sequences. And the amino acid sequences, once they change, the 3D structure also changes. Remember that the protein's function is directly correlated to their structure. If you change the sequence of the amino acids, you change the structure, thereby it affects the function. So yan siya.
Sir, we have 2 questions po sa Facebook Live.
Yeah.
No? Sagutin natin baka magtampo po sila. Ito po, a question po ni El Operario Mercado. Ang question po is there an existing legally binding agreement that's enforceable in the Philippines about GMOs or perhaps in the international community?
Yes. Wait lang, I'm trying to recall. So, eto kasi, the National Committee on Biosafety of the Philippines is a committee that oversees all of the concerns that possibly involves biosafety. And these include genetically modified organisms. And this particular body oversees, number one, the ethics of the study. And at the same time, kung ano yung maging epekto niya. So it also looks into the experimental process needed for the researchers to achieve whatever their objective is. These laws, or rather, the rules and regulations set by the National Committee on Biosafety of the Philippines is mirrored after international standards. And these standards include the Kyoto Protocol, the Cartagena Protocol, and these are international laws that govern how to deal with genetic manipulation. So the answer to that question is yes. Our local laws are pursuant to the international laws that we have. And truth be told, sobrang ayaw mag-work ng mga international scientists sa Philippines because we have one of the most stringent, one of the strictest laws when it comes to genetic modification and its mobilization. Kaya sobrang ayaw nila. But ayun, yes, our laws are pursuant to international laws.
Thank you, sir. Ito po yung isang question, sir, from Micaela Carpio.
Mhm.
Is there a high possibility that the genetic modifications would grow in the near future despite being irreversible, specifically to humans?
Genetic modifications to grow.
Would grow in the near future despite being irreversible. Kumbaga parang kusa po siyang mag-evolve ata or mag-change.
Okay. Note for a fact that genetic modifications happen in nature, even without human intervention. So yes, because of the mutagens that are present. Say for example, there's even a particular bacteria that can cause plants to mutate. These mutations come in the form of inserting their own genome to the plant itself, and that is Agrobacterium tumefaciens. We have viruses that incorporate their genomes into our own cells, making us also genetically modified in the sense. So the answer to your question is yes, genetic modification with or without human intervention is a thing in nature and it occurs naturally. So ayan. Ay, Sir Josh, may gusto akong sagutin na tanong dito.
Yes, sir.
Sir, GM foods can possibly cause development of disease which is immune to antibiotics? No. Kasi remember, say for example, balik tayo sa golden rice. Golden rice will only produce the proteins that is generally produced by yellow pigmented na crops. And that is and these are the genes for, or rather, these are coded by the genes for the beta-carotene synthesis. Hindi mo siya mako-correlate to antibiotic resistance if yung ipoproduce lang naman ng cell na yan is simply the genes necessary for, say for example, for the production of the yellow pigment, which is healthy, which is vitamin A for our eyes. So in short, development of disease, I think the answer to that is no. Kasi before being released into the market, very strict trials must be put to place prior to saying that the organism is safe for food and feed. And these are also being looked into, say for example, antimicrobial resistance or ano pa ba? Allergenicity, toxicity ng GMs na to, tinitingnan yan siya ng ethics committee and ng screening committee. Kaya kapag nailabas na yan siya sa market, I can bet my 10 fingers na it's safe for anything. There is no fear in consuming GMOs talaga. It's just that we have these corporations with selfish desires na, say for example, gusto nila na mag-flourish yung kanilang negosyo. But but in order for them to do that, they need to topple, let's say, for example, biotech giants. And they do this generally by bad marketing. And our generation is a product of that bad marketing. I grew up believing my high school teacher telling me na kapag kakain ako ng GMO, yung kaapu-apuhan ko tutubuan ng pakpak. Which is not the case. Bad marketing lang talaga, guys. And I do understand the stigma and the concern. But prior to jumping to conclusions, as educated members of this society, we should take it upon ourselves to gather enough information prior to us making the conclusions. Okay? So it's very nice to have these kinds of conversations so that things that need to be clarified are clarified. And thank you din for your honesty sa pagtanong.
Thank you, sir. Ito, I think I have one last question lang po
Yes.
on my chat box po. Ang question niya po, sir, is you have mentioned po na there will be times po na we can have a cancer-free society or HIV-free society, but will there thanks to genetically modified
Humans.
genetic modifications to be parang ganun po. Ang question niya po, sir, is will there be a potential ay parang may potential ba po na may ibang or mas isang paano ko ba i-phrase yung tanong niya? Parang may mas potent na virus perhaps. For example, tayo ang society natin HIV resistant. Will there be a potential na there will be another virus that will parang supersede that resistance dun sa virus na yon? Parang ganun, sir.
The answer to that question is yes. Pansin nyo, as our society progresses, the severity of the disease also progresses. This is because of the fact that, number one, there will always be co-evolution of host and pathogen. Habang gumagaling yung pathogen sa paglaban sa host, eventually the host will catch up. Host, pathogen, host, pathogen. So there will always be forever this existing arms race between the host, or rather, between the pathogen and the host, the pathogen and the host. So eventually, I don't know, say for example, there are billions of viruses out there. And I don't know when will be the next event that two viruses will come to meet and eventually produce a very, very bad pandemic. So, ayun siya guys, these things are not avoidable. We are always bound to experience new diseases from time to time. So the most that we can do is to accept this inevitability and prepare the country for it na lang. Kasi hindi talaga siya maiwasan, guys. These pandemics are bound to happen because such is life. Sabi nga dito ni John Lloyd Subtinente, GMO is one of the key to achieve Philippines food security. The answer to that question is yes. Kasi we live in a very harsh environment now. We are always battered with storms. With the advent of global warming, our crops are not heat tolerant and storm tolerant and drought tolerant. So the only way to induce this is to introduce genes that will enable them to be drought tolerant, heat tolerant, and stress tolerant. So ayun.
I think that's all for our questions, Sir Eman. So thank you very much, Sir Eman. And thank you to all our participants. This is a very fruitful conversation, no, on the applications of genetics and its advantages and disadvantages. Thank you very much, Sir Eman, for answering our questions. Thank you, po. So, all right. So at this point, Team LRC would like to take this opportunity to thank our speaker for his valuable time. So to award a certificate of appreciation, I would like to recognize our director here at the Learning Resource Center, Assistant Professor John Mervin L. Mbate. Allow me to first read the citation. University of the Philippines, Los Baños Learning Resource Center presents or awards a certificate of appreciation to Emmanuel T. Galang for serving as resource speaker during the GENE-ius Talaga, a tutorial webinar series on genetics during the third session entitled Parallel Minds, Applications on Different Fields of Study, held today, December 9, 2022, at the Learning Resource Center, UPLB College, Laguna, signed by our director, Sir J.M. Mbate. Sir J.M.
Thank you very much, Josh. And of course, Maraming salamat po Sir Eman sa pag-spend ng iyong 3 araw with us. No? Napaka-fruitful ng discussion natin. Ako, I myself am not an expert in biology and genetics, no? But I think our discussion today is really quite fruitful. Medyo na-demystify natin yung mga agam-agam natin about GMOs. But I think the beauty of this is that we're able to apply our abstract concepts, our research, our studies into something that is practical and something that could help our country progress and, you know, help alleviate poverty in all its forms. No? Kasi yun naman yung gusto nating mangyari. So sa mga teachers na nanonood sa atin ngayon, I hope na meron kayo nakuhang mga techniques on how to you know, teach genetics, genetic concepts to our students in a friendlier, more more understandable way. And for our students who are here, especially the UPLB students who are tuning into us right now, hopefully ay mas naging confident tayo to ace our examinations, our upcoming examinations, most especially that the finals week is just around the corner. At sana ay mas nakita natin yung beauty ng genetics dun sa pang-araw-araw natin na ginagawa. And Sir Eman, maraming salamat ulit doon sa pagpayag na maging speaker namin. Medyo sinulit ka talaga namin, no? And we hope na dumami pa yung blessings mo in life and excited kami doon sa journey na naghihintay sa'yo. Maraming salamat po ulit.
Thank you very much, Sir JM, and thank you ulit po kay Sir Eman. So gusto ko pa yung sabi ni Sir JM, no? So hopefully this 3-day seminar or 3-day tutorial webinar series on genetics will help encourage our students to pursue research on different aspects of genetics, no, to further expand, no, yung ating contributions dito sa field ng genetics dito sa ating bansa. All right. So thank you very much, Sir Jem and Sir Eman. Now for the evaluation of this session, please do not forget to answer the evaluation form after this tutorials to receive your e-certificate of attendance. So your comments and suggestions will be deeply appreciated by our team, #TeamLRC. So the evaluation link is being shown right now on your screen. So it's tinyurl.com, GENE-ius eval session 3. And I'll also put it here in our chat box so you can easily go to the form and kindly answer it to receive your certificate of attendance. And the deadline of responses is until 10 p.m. tonight. Okay? So we hope that we all gain inspirations and valuable insights from our speaker during this 3-day tutorial webinar series. At this point, we'd like to recognize and thank our team, #TeamLRC, and our GENE-ius Ka Talaga team, headed by our director, Sir JM Embate. On our technical side is our student assistant, Mr. Hans Benedict Aguilar. And a big shout out to Team LRC's members, Ate She, Ate Majeng, Tita Allen, and Kuya Iwo. And a big thank you to our speaker, Assistant Professor Emmanuel P. Galang, for being very passionate and inspiring for the past 3 days. All right, so 3 out of 3 sessions, dear students and teachers. Thank you so much for joining us in this 3-day tutorial webinar series on genetics, and we hope that you will join us in our future tutorials on other subjects in the arts and sciences. So please stay tuned. Allow me to end this session with a quote from the one and only Charles Darwin. So he said that it is not the strongest of the species that survives, nor the most intelligent. It is the one that is the most adaptable to change. So thank you, dear friends, dear teachers, dear students. Stay hopeful, stay healthy, and stay safe. We'll see you on the next tutorials. Thank you, everyone.
Maraming salamat. Maraming salamat.